Hi everybody, it's Dr. Lois White here and welcome to this final online lecture for nutrition. Today we'll be covering some of the micronutrients involved in energy metabolism. Micronutrients are often categorised into those with antioxidant function, such as vitamins A, C and E, those involved in hemopoiesis, such as the nutrients we've looked at over the last couple of lectures, those involved in bone health, such as calcium and vitamin D, and those involved as cofactors in energy production. That's not to say that there's not significant overlap. Remember last week we discussed iron's role in oxidative stress and energy regulation, for example. So there are many micronutrients involved in energy regulation. However, today we'll discuss the key nutrients that act as cofactors, and I've also included the DRVs for these micronutrients on the last couple of slides. So I've included a little red star next to the micronutrients we'll look at in more detail today. You'll notice that many of the B vitamins are included in this list. We'll discuss vitamin B1, also known as thiamine, vitamin B2, otherwise known as riboflavin, B6 or pyridoxine, copper, which is a trace element, and niacin, also known as nicotinic acid, which is part of the vitamin B family, along with nicotinamide. Some other micronutrients involved in energy metabolism include pantothenic acid or vitamin B5, which is actually named after the Greek word pantothen, meaning from all sides, as it's really widespread in foods and deficiency is actually very, very rare. Zinc and some other trace minerals are also involved in energy metabolism, and if you want to read a little more about these, they're covered in Chapter 13 of Maguire and Bierman's Nutritional Sciences from Fundamentals to Food. So let's have a think about what foods are rich sources of these micronutrients and actually what subgroups may be at particular risk of deficiency. So vitamin B1 or thiamine is found in whole grains, meat, peas, beans and lentils, and actually more refined foods tend to contain less thiamine unless they've been fortified. It's also worth noting that white flour is actually fortified with vitamin B1 by law in the UK. Vitamin B2 or riboflavin is found in milk, dairy products and cereals. Vitamin B3 is found in meat, particularly beef, milk, fish and whole grains, although it's worth noting that corn isn't a good source of vitamin B3. Vitamin B5 is found in eggs, whole grains and meat. Vitamin B6 is found in meat, fish, egg yolks, whole grains, bananas, avocados, nuts and seeds. Zinc is found in shellfish, but it's not something that we consume in large amounts in the UK. However, it's also found in things like ginger root and garlic, meat, peas, nuts, egg yolk, and whole grains as well. So there are lots of different sources of zinc. The copper content of foods will depend on the copper content of the soil that plants are grown in or animals feed on, but oysters, kidney, dried legumes, liver and nuts are all good sources. However, remember that liver and raw shellfish should actually be avoided during pregnancy. Chronic alcoholics are the main subgroup in the UK to suffer from vitamin B deficiencies, particularly B1, and we'll talk about this and why this happens a little bit later. But copper deficiency can occur in hospitalised patients or preterm infants receiving improper nutritional support due to its absorption in the stomach. And additionally, vegetarians and vegans may be at increased risk of zinc deficiency, as zinc, in a similar way to iron, is less bioavailable in plant sources. So let's take a closer look at vitamin B1. There are several forms of the vitamin, thiamine triphosphate, diphosphate and monophosphate. However, the main active form of the vitamin in the body is thiamine diphosphate, otherwise known as pyrophosphate. This is essential for energy metabolism and DNA synthesis. In healthy adults, around 25 to 30 milligrams of pyrophosphate is found in the body tissues at any one time. And as we've previously discussed, thiamine is water soluble, so it's not stored in the body. Each day, the amount needed has to be consumed, or functionality will not be optimal. So in the small intestine, phosphatase groups are unbound from thiamine by phosphatase enzymes. Absorption occurs primarily in the duodenum via active transport, which is a sodium-dependent ATPase mechanism in the case of thiamine. Active transport will work up to saturation level, and thereafter passive transport occurs. However, this is actually ineffective, and any remaining thiamine is excreted in the urine. Alcohol actually inhibits active but not passive transport, hence why alcoholics can easily become deficient in B1. In terms of metabolism, the thiamine is now rephosphorylated to TPP, the active form of thiamine, and is transported freely in the blood plasma. However, it can also be transported bound to albumin, and thiamine is excreted in the urine. So let's look at the functions of TPP. TPP acts as an essential coenzyme in several key reactions in metabolism. It's required for the activation of the enzyme that converts pyruvate to acetyl coenzyme A. Acetyl coenzyme A participates in many biochemical reactions in protein, carbohydrate, and lipid metabolism, and its main function is to deliver the acetyl group to the Krebs cycle to be oxidized for ATP production. TPP also acts as a coenzyme in the conversion of succinyl coenzyme A from alpha-ketoglutarate in the Krebs or TCA cycle, and it's involved in trans-ketolase reactions in the pentose phosphate pathway. Remember, the pentose phosphate pathway generates NADPH and pentoses, which are 5-carbon sugars, as well as ribose 5-phosphate, which is a precursor for the synthesis of nucleotides. So the turnover rate of thiamine is high and deficiency can occur within 21 to 30 days. The initial response to low intakes of thiamine is to reduce renal excretion. This will slow but will not stop completely. Lethargy and muscle weakness can occur due to reduced pyruvate dehydrogenase activity, which is activated by thiamine. There will be reduced activity of the TCA cycle and the pentose phosphate pathway. You can imagine if you're not making enough ATP or nucleotides for DNA synthesis, then your energy levels will be very low. However, it's important to note that these symptoms are common to many deficiencies and diseases. Weight loss occurs because the body will look for energy elsewhere. The body assumes that there are no substrates with which to make energy from, so macronutrients stored in the body start to get broken down for energy. But this actually won't help, as it's the cofactors that are stopping the system working. It's a little bit like having a steam engine full of water but no coal to heat it up. Neurological effects can occur as acetyl coenzyme A is a precursor for the neurotransmitter acetylcholine, which is a chemical released by nerve cells to send signals to other cells. Additionally, lactic acidosis can also occur where your blood pH drops. This is because the body can't convert pyruvate to acetyl coenzyme A, so it gets converted into lactate instead, which is an anaerobic pathway. So there are actually 2 deficiency diseases associated with thiamine. Firstly, beriberi. This means extreme weakness, or literally I can't in Sinhalese, and was actually prevalent in World War II prisoners of war in Asia due to poor dietary intakes. And it occurs due to a prolonged thiamine deficiency. And risk factors include a diet of mainly polished white rice, alcoholism, dialysis, chronic diarrhea, or taking high doses of diuretics. Now, there are 3 main classifications of beriberi. Wet beriberi involves severe edema in the arms and legs, shortness of breath, and enlargement of the heart and respiratory issues, which can result in heart failure. Dry beriberi is found mostly in adult populations and results in neurological effects such as lack of feeling or tingling in the feet and toes, leg cramps, and muscle wasting. And infantile beriberi can occur in breastfed babies whose mothers are deficient in thiamine and can lead to cardiac arrest in some cases. Wernicke-Korsakoff syndrome, or WKS, is usually secondary to alcohol abuse because alcohol reduces absorption of thiamine and only 20% of cases are identified before death. This failure in diagnosis of WKS and thus treatment of the disease leads to death in approximately 20% of cases, while 75% are left with permanent brain damage. It causes ischemic damage to the brainstem and its connections. Encephalopathy does not refer to a single disease, but rather to a syndrome of overall brain dysfunction, and in this case includes ataxia, which is a lack of voluntary coordination of muscle movements that include gait abnormality, and vestibular paralysis. The vestibular system includes the parts of the inner ear and brain that process the sensory information involved with controlling balance and eye movements. And WKS is also associated with confusion and short-term memory loss. Okay, so let's move on to vitamin B2. Vitamin B2, or riboflavin, is named after its ribose component. The body uses riboflavin to produce two coenzymes, flavin adenine dinucleotide, or FAD for short, and flavin mononucleotide, or FMN for short. FAD is the main form, and approximately 1 gram is found in the liver and kidneys. So let's talk about digestion and absorption of vitamin B2. So FAD and FMN are released from proteins by gastric acid in the stomach, and they're actually bound to proteins in food as they're more stable in this way. In the small intestine, phosphatase enzymes hydrolyze FAD and FMN to leave free riboflavin, and this is because only free riboflavin can be absorbed in the small intestine. And absorption actually occurs in the small intestine in exactly the same way as thiamine. So riboflavin is transported freely in the blood plasma, and as with thiamine, there's no storage capacity for vitamin B2. So similar to vitamin B1, riboflavin is involved in formation of a number of enzymes mainly found in the liver. And these enzymes are involved in removing molecules of hydrogen and allowing oxygen to attach, so these redox reactions. As with thiamine, FAD acts as a coenzyme in the conversion of alpha-ketoglutarate to succinyl coenzyme A in the TCA cycle. It acts as a coenzyme for succinate dehydrogenase in the conversion of succinate to fumarate in the Krebs cycle, and again in oxidative phosphorylation in the electron transport chain, which leads to production of ATP from ADP. FAD is also required in the conversion of vitamin A, vitamin B6 and folate to their active forms in the body. So deficiency of riboflavin is rare. However, the vitamin B family tend to be found together in the same foodstuffs. So a deficiency of one is often a sign of other vitamin B deficiencies. And deficiency, as with thiamine, is also associated with alcoholism. In terms of symptoms of deficiency, in adults you often see atrophy of fast turnover tissue such as the skin, mouth and tongue, and these can become sore and inflamed. And because vitamin B2 is involved in folate metabolism, it can also lead to anemia, and it can result in stunted growth in children due to its involvement in energy and vitamin A production. So as previously mentioned, niacin is the generic name given to a range of forms of vitamin B3. However, the two main forms are nicotinic acid and nicotinamide. The main active form in the body is nicotinic acid, and we can actually synthesize niacin in the body to a certain extent from the essential amino acid tryptophan. 1 milligram of niacin can be made from 60 milligrams of tryptophan, and therefore foods containing niacin or tryptophan are considered dietary sources of niacin. Again, niacin in animal products is more bioavailable than in grain foods, as niacin in grain foods is bound to proteins, making it less available for digestion and absorption. The main form of niacin in the diet is nicotinamide adenine dinucleotide. NAD is the oxidized form and NADH is the reduced form. Cooking or processing can release the free forms of niacin, or enzymes within the intestinal mucosa hydrolyze NAD or NADH to release the free form. A small amount of niacin is absorbed in the stomach, but it's mainly absorbed in the small intestine in the duodenum, and it's absorbed in exactly the same way as vitamins B1 and B2. So nicotinamide, which is the main form in the plasma, then circulates in the blood to the liver where it's attached to transport proteins.
It's then released by the liver to cells that require it. NAD and NADH, i.e. the coenzyme forms of niacin, can be synthesized from both nicotinamide and nicotinic acid in most tissues. The amount of niacin in the liver is constantly being used. It's a pool of niacin rather than a store, much like the liver's protein pool. And niacin excretion is minimal due to really efficient glomerular reabsorption in the kidneys. Like vitamin B2, niacin-derived coenzymes, NAD and NADP, act primarily as hydrogen acceptors and donators in redox reactions, both in energy metabolism and metabolism of fats and DNA. Some studies have shown that consuming large amounts of niacin could actually lower LDL cholesterol and raise HDL cholesterol levels. However, we know little about the mechanisms behind this, and actually taking large amounts of niacin in supplement form could damage the liver. So deficiency of niacin is rare because we excrete very little. However, if deficiency does occur, it can lead to general lethargy, disorientation, a swollen mouth and bright red tongue, again because fast turnover tissues are going to be affected first. And because we start using more tryptophan to make niacin, tryptophan is diverted from serotonin synthesis, which can actually lead to depression. The deficiency disease associated with niacin, pellagra, is seen mainly in lower income communities in Central and South America, where corn, otherwise termed maize, is the main food source. Chronic high alcohol consumption or low protein consumption, i.e. low tryptophan intake, can also lead to deficiency. So long as there's adequate protein in the diet, we can convert tryptophan to niacin. So deficiency disease is mainly seen in famine situations. Symptoms of pellagra are sometimes referred to as the 3Ds. Diarrhea occurs due to damage in fast turnover tissue in the GI mucosa. Dermatitis, again, is associated with fast turnover tissue of the skin. And dementia can occur due to the rerouting of tryptophan. So let's look at vitamin B6. Vitamin B6 can be found in many forms, including pyridoxine and pyridoxamine. However, the most active form found in the body is pyridoxal phosphate, and it's a coenzyme form of the vitamin. It's found in all metabolically active tissue. However, all forms of vitamin B6 have similar biological activity in the body and are involved in over 100 chemical reactions. Pyridoxal phosphate is also the most common form found in food sources. However, it undergoes dephosphorylation by alkaline phosphatase prior to absorption in the jejunum. Absorption occurs directly through passive diffusion, so without the need for energy like some of the other B vitamins. Once absorbed, vitamin B6 circulates in the blood to the liver, where it's rephosphorylated to PLP. PLP is then bound to enzymes in its role as a coenzyme. There are no stores of PLP in the body, however, relatively large amounts are found in the liver and muscle cells bound to the enzyme glycogen phosphorylase. Remember, glycogen phosphorylase breaks down glycogen into glucose in these tissues. Excess PLP is dephosphorylated and converted to 4-pyridoxic acid for excretion in the urine. Remember that I previously mentioned PLP acts as a coenzyme in over 100 chemical reactions in the body, including reactions in protein and carbohydrate metabolism. Examples include the conversion of glycogen to glucose-1-phosphate in glycogenolysis, i.e. the breakdown of glycogen stores in the muscle and liver tissue to create glucose for energy. It's also involved in transamination and decarboxylation of amino acids. These reactions are required for the synthesis of non-essential amino acids from essential amino acids. Without vitamin B6, all 20 amino acids would be essential. Vitamin B6 is needed for the synthesis and function of hemoglobin and also acts as a cofactor in the conversion of tryptophan to niacin. So vitamin B6 intake is very important if niacin intakes are low. So alcohol abuse can impair absorption of vitamin B6 in the small intestine, which may also result in niacin deficiency due to reduced synthesis of niacin. Additionally, due to B6's role in hemoglobin synthesis and function, deficiency may lead to a condition called microcytic hyperchromic anemia, where the concentration of hemoglobin in red blood cells is low. This reduces the ability of the red blood cells to carry oxygen to cells and their ability to produce ATP via aerobic respiration. The red blood cells are small and pale under an electron microscope in the case of microcytic hyperchromic anemia. So for the last part of the presentation, I'm going to focus on copper. Copper is an essential trace mineral vital to health of all organisms, and it acts as a coenzyme in a wide variety of processes. It's important in red blood cell production, ATP production, nerve cell function and immune system function. And I think it's also worth mentioning that copper and iron are found in many of the same food sources. In adults without deficiency, there's around 80 to 100 milligrams of copper in the body, 40% is found in the muscle, 15% in the liver and 10% in the brain tissue. So copper does not require any digestion, and it's absorbed in both the stomach and the small intestine via active transport and passive diffusion. Excessive intake of iron can actually reduce the bioavailability of copper because they compete for the same transport mechanisms. Also, heavy antacid use can reduce bioavailability as it forms insoluble complexes that are not readily absorbed. As with iron, copper absorption increases when status is low and decreases when status is adequate. Absorbed copper is transported to the liver, where it's bound to a number of transport proteins. However, ceruloplasmin is the major copper-carrying protein in the blood, and this protein also plays a role in iron metabolism. Free copper in the body is highly reactive and toxic as it drives the production of free radicals that cause oxidative damage to cellular structures. So due to its toxic potential, copper is normally tightly bound to copper-carrying proteins inside the liver, and excess or free copper is eliminated from the liver via vascular excretion into bile, so it's excreted in the feces. So here's a nice diagram of copper metabolism just for reference. It's worth mentioning that only around 25% of dietary copper is absorbed, 25% is lost directly in the stools, and 50% forms a complex called metallothionein, which is also excreted in the stools. Less than 5% of copper circulates in the bloodstream, and a very, very small amount of this is free copper due to its free radical activity. And although most copper is excreted in the stools, less than 5% is excreted in the urine as well. So copper acts as a cofactor for metalloenzymes, which are involved in redox reactions in many metabolic pathways. Ceruloplasmin is also involved in transporting iron to sites of erythropoiesis, i.e. the bone marrow. Copper acts as a cofactor for the enzyme cytochrome C oxidase, which combines electrons, hydrogen ions and oxygen to form water in the electron transport chain. Thus, copper is vital for ATP production. Copper is also used in synthetic enzymes to promote synthesis of neurotransmitters and collagen. So copper deficiency is rare, however secondary deficiency may occur due to protein energy malnutrition, for example, or persistent diarrhea and vomiting, and prolonged copper deficiency can lead to bone weakness and anemia. And as copper absorption occurs in the stomach as well as the small intestine, patients receiving enteral nutrition via a jejunostomy tube may require copper supplementation intravenously to avoid deficiency. So the next few slides just outline the DRVs for both the B vitamins and copper in children and adults. I hope you have a better understanding of how vitamins and minerals play a role in energy metabolism, and do feel free to email me if you have any questions about what's been covered in the lecture today.